And now, from San Francisco and the UCSF Rosenman Institute, the Health Technology Podcast with your host, Christine Winoto.
Imagine discovering your child has a fatal disease before they're even born. Now imagine there's a chance to cure it before birth using cutting-edge gene therapy or fetal stem cell transplants. That's the world Dr. Tippi MacKenzie is helping to build. A pediatric and fetal surgeon, director of the UCSF Broad Stem Cell Center, and trailblazer in utero treatments for congenital disease, Dr. MacKenzie's work feels like science fiction becoming science fact. But before she was a surgeon, she was a Juilliard-trained pianist. Her journey from Bach and Brahms to stem cell and genome editing is a fascinating one. And in this episode, she shares it all. We talk about the unique immune tolerance of the fetus, the ethical and scientific frontier of gene editing in utero, and her vision to transform fetal surgery into fetal genome surgery. She also shares the story of how an article in the New England Journal of Medicine changed the course of her life, and how we can better support the next generation of scientists and physicians innovators. In our conversation, you'll hear how Dr. MacKenzie is bridging science and surgery to give babies a healthy start before they're even born. Here's our conversation. Welcome, Tippi. Thanks for joining today.
Thank you so much, Christine. Lovely to be here.
It's so nice to see you. And we had you a few years ago at our symposium where you talk about the work that you did. And it's almost like science fiction for me, at least the first time when I heard that. But before we start, I always like to start with our guests is their journey. What took you to be interested in healthcare? And I think you mentioned to me before that you were a pianist. And it always tickled me that you're a pianist, yet you're doing what you're doing now. So.
Oh, thank you for asking. Yeah, so I grew up playing the piano. It was a big part of my life growing up. And I love classical music. I mostly the three Bs, honestly. I love kind of deciphering, Bach toccatas and French suites and whatnot. And I grew up thinking I was going to be a pianist. I went to Juilliard pre-college before college. And then something in me also said, I really like science and math, and I didn't want to go to conservatory and be limited to only music. And so I went to college and I ended up majoring in biochemistry. And I really loved what I was learning. I loved learning about the body, and I loved learning about the cell biology and all the different processes. And I didn't want to actually go to medical school. I thought I was afraid of blood, which is funny to say now that I ended up becoming a surgeon, right? In college, I worked in a lab, and I thought that the lab stuff was really interesting, but somehow I didn't have a great experience working in lab. And I thought, oh, okay, well, what else do you do with a biochemistry major? I guess I'll go to medical school, even though I'm afraid of blood. Maybe I'll be a psychiatrist or something. And I tell the story to trainees because it's just it's evidence that you don't necessarily know yourself when you're, during those formative years, and you just kind of need to keep your eyes open and your options open. So I ended up going to medical school. I went to Stanford Med School, and literally one of the first things I did was to find a lab to join. I joined Robert Sapolsky's lab. He's this amazing neuroscientist, and he had a piano in his lab. And so it was a place to both be a scientist as well as we would have talent shows. And I loved the research in his lab, and I loved medical school. I loved once I actually got into the wards and I did a surgery rotation, and I could see just the immediate impact that had on people's lives. I left thinking, okay, I'm going to be a surgeon. And one of those days on my surgery rotation, I heard about fetal surgery. I heard that, there was a Dr. Harrison up the road at UCSF who had started operating on fetuses in the womb. And I thought, wow, that sounds amazing. And maybe I could go and do a rotation there. I'm like a third-year medical student. I don't know anything, but it was the days before email. So I literally called up his office and somebody answered the phone and was very helpful. And in a couple of days, I was on his service doing just kind of like following him around like a puppy dog. And they did a fetal operation, which is, they're rare, but I just got really lucky. And I remember standing in the corner of that room watching him do this. And I thought, this is amazing. That's what I want to do when I grow up. And also that year was the first report in the New England Journal of a fetal stem cell transplant. So I came home from a run one day and I saw, and, I used to get the New England Journal, like the print copy, right? So I saw this article and they had cured a child with severe combined immunodeficiency, which is the bubble boy disease, by doing stem cell transplants before birth. And I was like, wow, you can do this complex fetal surgeries and you can do the stem cell transplant. So you can like do the surgery and the science, and, what an amazing field. So, that's kind of a long-winded story, but it really, those were the influences on me, during my formative years. And, I still feel the same way about just the amazing, opportunities and in this field, in surgery and really healthcare in many forms.
Yeah, I mean, Dr. Harrison did a great job in recruiting you here, and he is amazing.
He's an
individual too. So it's
a true innovator.
Yeah, and so tell us a little bit, about the stem cell and the fetal surgery. I guess maybe to some of us who don't know, like, why is it better? Why is better to do it when they're still in the fetal to do the stem cell?
Yeah, great question. So fetal surgery was started, by Dr. Harrison, as we said, to treat fetuses who are going to die otherwise from their underlying disease. And it got started for anatomic conditions that you could see by ultrasound. So big tumors or, tumors on the lung, tumors on the back, diaphragm problems. But then people immediately saw this other opportunity, which is that the biology of the fetus is really unique. And one of the elements that make it really unique is that the immune system, while it's forming, it's more tolerant of new proteins. Because think about it, the fetus is learning to tolerate its own self. It's learning to tolerate the mother's cells, which are crossing back and forth into the fetus. And it's learning how to, early after life, it's learning how to tolerate the microbiome and other organisms it encounters. So it's a really unique window of opportunity to introduce new cells or new proteins without mounting an immune response. Whereas we know that if you do, say, a stem cell bone marrow transplant after birth, you have to do immunosuppression and you have to give all kinds of medicines that can, increase the toxicity of that. Back in the 1950s, Billingham, Brent, and Medawar did these Nobel Prize-winning experiments where they infused stem cells from one strain of mouse into another during fetal development, and they saw that they could induce tolerance, to that, donor strain of mouse. And so that, that sort of idea that this could take place has been around since the 1950s. And that article I read when I was a medical student where they treated the boy with bubble boy disease, that was a stem cell transplant that cured that little kid. So there's been this idea of the opportunity, but what's been really fascinating is that there's a lot more science to be learned, and the field really hasn't achieved its full potential until we were able to really understand, like, what is the mechanism? Why is the fetus tolerant? What kinds of immune cells are there or are not there? So, and it's still a field in which there's a lot of discovery to be done.
How much do we know now in terms of how it works, why it works?
We know quite a bit. So there are these special immune cells called regulatory T cells that suppress an immune response. And they evolved in placental mammals, probably to promote tolerance between the mother and the fetus. We know that pregnant mothers develop regulatory T cells that protect against an immune response against their fetus. We also know that fetuses develop these regulatory T cells to protect against an immune response to their own, proteins, as well as to maternal proteins. And that knowledge actually came out of Mike McCune's lab around the time I started at UCSF. He had a paper in 2008 that really described that for human fetuses. They're developing regulatory T cells against their own mothers. And that's really part of the magic of how pregnancy works. Both the mother and the fetus have to actively tolerate each other. It's not that, they don't see each other, there's this impermeable wall. It's not that at all. It's active tolerance by these very special cells. And that was a really important clue for the whole stem cell transplantation field, that if you want to do a successful stem cell transplant into a fetus, you need to give stem cells from the mother, because the fetus already has learned how to tolerate those cells. So after that New England Journal paper, a lot of people had tried to do stem cell transplants with somebody else's cells, thinking, the fetus is tolerant, we can do this. It doesn't work unless you give the mother's cells.
It's tolerant to specific person.
Yes.
Not just to anybody.
Yes.
It sounds like that with the potential of the impact of this is that babies that were born would be all come out healthy.
That's the goal. That is the goal. I mean, we're a long way from there. There's a lot we need to do to sort of tweak how well stem cell transplants work. We did a trial a few years ago where we did give maternal stem cells to fetuses with another disease called thalassemia, alpha thalassemia. And we got some engraftment, but not enough to be curative. And so the barrier we came up against was that those fetuses are sick. So their own stem cells multiply so much so that there's no room in their bone marrow for the stem cells we're giving. So even if they're not per se rejecting those cells, there's just not enough space. So it sort of sends you back to the drawing board, back to the lab to try to figure out, what can we do to kind of wipe out the fetus's own bone marrow to create space for our cells that we're giving? And how do you do that safely, without causing toxicity, especially if the fetus is already sick? And so, you're sort of limited on how much you can do. So all of that is, it's really exciting. And it just also teaches you, for every one thing you think there are 10 other things you need to understand. And they're really the vital importance of the sort of basic biology research to understand the black box of fetal biology so that we can, really develop therapies so that, yes, our goal, our motto is so that, every child can have a healthy start in life.
Yeah. In the bubble boy disease, now that if you got diagnosed as a baby, now it seems to be doesn't need to be in existence again, right? But
yes.
You're saying about the thalassemia one, we're not quite there yet.
We're not there yet, yes. The bubble boy just turned out that it was probably the easiest disease to get it to work in, because there is no immune system, but also the bone marrow is more empty. So, stem cells engraft better if you're immunodeficient. So, yeah, so it's been 18 years of, having my lab here at UCSF now, and there are always, questions to answer and amazing colleagues to try to answer these questions with. And it leads us in new ways, in gene therapy and genome editing. If, for example, the thalassemia, the alpha thalassemia, okay, if it's really hard to engraft the stem cells because the bone marrow cells are multiplying like crazy, well, that's a really good starting point to do genome editing, because you want the cells to be dividing for them to be editable. So that's almost the perfect setting for a different solution. So you kind of have to be nimble with the armamentarium you have of the kinds of therapies that you can offer. So if one doesn't work, you develop another one.
Is that happening now? The gene editing happening in the fetus stage?
In mice. That's what we're working on. That's what we're working on in mice and also in patient cells. So one of the most amazing things about being a clinician scientist is that you can do sort of patient-facing research. And what we learned from our clinical trial is that, yes, the stem cell transplant maybe isn't the best kind of treatment, but genome editing might be. And we've developed relationships with not just the patients who have been in the clinical trial, but the international community of patients with alpha thalassemia who have kind of been ignored because the whole, the medical community has said, oh, this is a fatal disease, there's not much to do. But as we sort of shine the light on it, we see that actually there are lots of children living amazing lives who desperately need treatments. And, in the industry and in the medical field, nobody has bothered to develop a gene therapy or genome editing strategy for them.
Mm-hmm.
And so we're doing that now.
For some of us who don't know, can tell us a bit about the alpha thalassemia and what usually are the prognosis? What's causing it?
Yeah, great question. So alpha thalassemia is sort of the cousin to beta thalassemia. So it's a red blood cell disease. We all have this molecule called hemoglobin in our red blood cells. Red blood cells carry oxygen, and hemoglobin is the molecule that carries that oxygen. And hemoglobin is made up of 4 different chains that are linked together. 2 of them are alpha, 2 of them are beta. So if you have beta thalassemia, you're missing your beta chains. If you have alpha thalassemia, you're missing your alpha chains. It just so happens you need your alpha chains when you're a fetus, whereas you don't need your beta chains yet. And so alpha thalassemia fetuses get sick. And that's why the dogma in the field has been, oh, they get sick and then they died before birth. But, we're in the age of fetal therapy. They've been getting blood transfusions to survive. And if you just get a couple of blood transfusions, which we do all the time for other anemias, they do just fine. They have normal neurologic outcomes. They end up looking like a child with beta thalassemia where they need a blood transfusion once a month. So yes, it's a chronic disease, but it's not as burdensome as many of the other chronic diseases that we see in our, fetal and pediatric population. What we're trying to do is to develop a genome editing or gene therapy for them that would be given either before birth or after birth if you can get them to birth with blood transfusions, which more and more people are doing now.
Mm-hmm. Is there other diseases that now is being tried to be cured with this fetal transplant, stem cell fetal transplant?
Yes, so there's been, I think a lot of us are really moving to the gene therapy approach because of this, unless you're a fetus with an immune deficiency, we need to create space in the bone marrow in order for those cells, even if they're derived from your mom, to engraft. So in a child, if you're going to do a bone marrow transplant, you always wipe out the bone marrow to create space for your new cells that you're putting in. In a fetus, we don't quite know how to do that yet. There are some ideas. People are developing non-toxic ways of wiping out the bone marrow, but we're not there yet to do it clinically. However, in the meantime, the gene therapy and the genome editing field has grown exponentially. I mean, if we think about, genome editing, right, was discovered by, Dr. Doudna and Dr. Charpentier, 10, 12 years ago, and there's already an FDA-approved genome editing strategy for sickle cell. So if that exists, that could be a lot more impactful than wiping out the bone marrow and doing a stem cell transplant.
Mm-hmm. And which brought me to, I just learned today you mentioned that you're heading the Stem Cell Institute here at UCSF. Tell us a little bit more about that and what is the goal and the mission.
Thank you. Yeah. So this opportunity came up about 4 years ago. I was chosen to direct our stem cell institute. We're the Eli and Edith Broad Center for Regeneration Medicine and Stem Cell Research. So the Broad family started 3 separate stem cell institutes in California, one at UCLA, one at USC, and one at UCSF. And so we've been in existence for 20 years. Our labs are on a beautiful hilltop building, the Dolby Regeneration Medicine building. And so we have about 25 labs in the building, and we have more than 150 affiliate members across all the UC campuses, doing research ranging from the basic stem cell biology, developmental biology, like how does an embryo form, how do all the different stem cells know where to go in the embryo, induced pluripotent stem cells, to more translational research using stem cells to treat diseases ranging from epilepsy to Alzheimer's disease to diabetes to hematopoietic diseases. So it's an incredible opportunity to set the vision for this basic science institute. And we're going through a year of strategic planning right now, and what we've all agreed on is that this is the time now to add to our expertise in stem cell biology and incorporate the genome editing of stem cells to develop therapies for patients. And so our tagline is Discover. So that's our basic stem cell biology, engineer, and heal. And so, some of us are all along that spectrum. Some of us do all of it. Some of us do, one end or the other. But what we're all understanding is that it's really important to have the perspective of how that pipeline develops, because any therapy, as we mentioned, relies on a really fundamental understanding of how the biology works. And so genome editing of stem cells, if that is to work, we have to really understand fundamental stem cell biology.
Yeah, and that's really cool. And I feel as you progress with time, you see all this interesting, exciting things that's going on in science, and things that you saw in the Star Trek start to come to fruition. Sometimes it's like, wow, you can do that now, not in TV, but in real life.
Exactly. In real life, yeah.
Yeah. Oftentimes, I always tell people is that a lot of the work that we see at UCSF is that it would be great if you never see it, because when you see it, because you're not healthy. So tell us a little bit more. I mean, right now there's a lot of challenges in science getting funding, and sometimes people don't think about like what's the impact, because hopefully you never have to use it, which not true. Most of the people that benefit from the science when you're in healthcare. But is there something that give us some, window why your work matters to the regular people like us who are not in science?
Oh my gosh. Yes. Thank you for asking that question. I mean, I think the work matters not only because, if we're sick, but I think, as human beings who have understood so much of our natural world, I think it's really important for us to understand how our bodies work. How do we develop? How does an embryo develop? Like, we were all embryos once. And we have to do a better job, I think, of educating the public and educating our children about the vital importance of stem cells, even if you never get sick. Isn't it amazing to know about how you can stay healthy and how you can? A lot of our investigators work on aging. We all age, right? It's even if you're not sick, of course, we're all going to get sick. How do you keep your health span, your healthy living ages, as long as possible? Many of us will want to reproduce. How the biological clock is a function of ovarian biology, right? The stem cells in your ovaries get used up. And guess what? When they get all used up, you start to age. As someone who's going through that now, it's kind of like night and day, like, how quickly it starts to happen. And all of that is really important to understand. And so the basic science research that is, in danger right now, is vital for our understanding of the natural world as well as human health. And what I see is, not only is it important for the, understanding of the biology, but it's the training of the next generation of scientists. And that's one of the most heartbreaking things about it for me is, our training grants, our students, our graduate students, our postdoctoral fellows, not only is their funding cut off, but, some of them are not able to find jobs because there are hiring freezes, or just the whole morale around becoming a scientist and what an amazing profession it is. I think it would be sad if that were lost because of potentially short-term gaps in funding.
I think the news definitely pointing that out, and sometimes it's hard to believe it when you don't see it. And of course, there's a joke that, not a joke, I think this is what happened. You get all your PhD in science, you end up working for investment banking because you got all the training, but then doing something completely different. And I think we do need to attract more people to stay in science, but people have to be have the opportunity for them to chase it, right? Because it's at the end of the day, you have to make a living.
Yes, and it can't be such a pyramid system. We can't, all the PhD students that we're training, they need to see that at the end of that long road of training, there will be, multiple different kinds of jobs, right? Not everybody has to run a lab in an academic center. There should be multiple kinds of jobs that allow you to contribute what you've learned so that you can, live your best life in the way that you want. And there's a place for all of us. I think one of the things that, we are finding out more and more is that, academia needs to partner with industry in order to bring these therapies to patients. And that's a place, obviously, in which your leadership has been so helpful to UCSF faculty, and sort of bringing that more and more into the way that we train graduate students so that option is on the table from day one, and we can train them for those kinds of either industry jobs or jobs in which they link academia and industry.
Do you have, because many, many years ago, there's the academic purist fate. Everything has to be. If you're working with the industry, it seems like you have been tainted or corrupted by the promise. And as we know, in delivering solution for healthcare, it takes a lot more than basics, in academia. What are the things that, do you think it changed because it has to change, or do you think the solution, the problem is so much bigger than a long time ago that you need more people collaborate more now than before? Because people don't need to change unless they have to change.
Yeah, it's a great question. I think one of the things also is that the opportunity is so much bigger now. I mean, we are living in an astounding age, where our capabilities, our diagnostic capabilities, I mean, think about genetic disease as something I think about every day. We can sequence anyone's genome in a matter of days. And if you have a genetic disease, we know exactly where's the typo in your 3 billion, base pairs. And we can theoretically design a genome editor to fix that using tools that exist today. What we can't do is manufacture that medicine in an academic lab, right? It has to be made in a specific kind of facility. It has to match regulatory standards, et cetera. So I think people are understanding that if you want to take your work and with its astounding capabilities to treat a human being, you need to collaborate. I mean, the opportunities are bigger than any one person and any one, even a small team of individuals can do. So we all need to evolve in order to really seize the moment of the possibilities that are afforded by the privilege of living in this incredible period in humanity's history.
I have to pinch myself sometimes to remember how amazing in the world that we are currently living. I mean, there's always, something that's bad thing happen in the world, but I think at the same time,
yes,
there's so much progress, right? That
Yes.
I joke, it's like, oh, in the old days, they would chop my head off if I did not do something right. And now at least, there's some more law to it. I feel my bar is so low. But what I'm trying to say that sometimes the news make it sound like the sky is falling, which is some part it is. I think, we don't want to let the sky completely fall. I think it just need to try to change it when we move the wrong direction. I always tell my kids, I said, like, if you go down the hill, when it's too far down, it takes a long time to climb back up. And climbing is a lot of work, right? And so if you know you're going to the wrong direction, just go back up quickly. Then at least you have not, spent that much time. One of the things that you mentioned earlier about the future generation, that training this future generation, what are the things that you do here at UCSF in terms of mentoring the younger generation, the future Tippi, fetal surgeon, also researcher?
Thanks for asking. Yeah, we do a lot. We can probably even do more. We have a lot of programs ranging from, high school to college internships, internships in labs where they come and they do like hands-on work. We try to reach out to students from underrepresented communities at both the high school and college and graduate student level. We have an amazing program called Propel that's run by Todd Neistel that helps trainees get positions in labs and then get into graduate schools and medical schools. We host interns internships that are funded by CIRM, the California Institute for Regeneration Medicine, master's students from San Francisco State University in labs so that they can get hands-on experience. And it's not just experience, but it's also, seeing the possibilities of what's out there. I think a lot about, for me, it was, I had no idea what it would be like to be a doctor. And I thought, oh, I can't stand blood, therefore I shouldn't be a doctor, right? I mean, I never knew any. And so you have to see it to be able to imagine it when you're so young. And I think, we're trying to provide those opportunities and the mentorships, not just by faculty, but sort of peer mentoring by students at their level or maybe a couple of years ahead to like really take them under their wing and teach how to do an experiment, how to keep a lab notebook, how to present your work at lab meeting, how to write an abstract, how to present at a conference. All of those are really important skills in addition to how to think like a scientist. And do you think that this is interesting and exciting for you?
Last question, I know we're running out of time here, is what are the exciting things that you're working on right now?
So really thinking about how can we turn fetal surgery into the field of genome surgery, right? How can we take now that we're diagnosing fetuses with a range of genetic diseases, because we have a prenatal sequencing program that a lot of families take advantage of, if we have diagnosed your specific typo, how do we design and construct a medicine for your baby so that it can be given either immediately after birth or in the future before birth? So that they can start life healthy before all the damage of their early onset disease has set in. So really excited about bringing that to patients as kind of the next step to the fetal therapy trials that we are doing right now.
So it is almost happening right now. How far away?
Well, we're I don't know how many years away it is. What we are doing right now is a clinical trial in lysosomal storage disease patients, and we're not doing genome surgery. We're just giving the enzyme that these fetuses are missing. We're giving the enzyme before birth. We've treated 6 patients so far. We've seen some benefits. And we see that there is a need there to do something a little bit more definitive, so that they don't have to keep getting this enzyme replacement therapy every 2 weeks for the rest of their lives. So, we're working in animal models, delivering genome editors using lipid nanoparticles and other materials. And we're getting really exciting results. But we're definitely not in patients at this time.
Yeah.
But I'm excited, you have to have a goal to move towards,
definitely.
right? And we have to do it safely. We have to do it ethically. We have to, follow all regulatory and ethical standards. So all of that, keeps us busy.
It's a really, I don't have the right word for it. I just, when I try to explain it, it's like, you're doing a lot of cool thing. That's what I always try to explain to people. Like, this is doing all the cool thing. That's what I was thinking about, the work that you're doing. So,
thank you, Christine.
We appreciate your work.
Thank
you so much for sharing your story and all the amazing work that you do.
Thank you so much, Christine.
Thank you for tuning into today's episode of the Health Technology Podcast. We hope you found the conversation insightful and inspiring. If you enjoyed this episode, please take a moment to subscribe, rate, and leave a review on your favorite podcast platform. Your feedback helps us continue bringing you the latest in health innovation. Stay connected with us on LinkedIn and Twitter for updates on upcoming episodes and more. Until next time, keep pushing the boundaries of what's possible in healthcare. I'm Christine Minato, and thank you for listening.